Hello friends, welcome back to my channel Plant Pathology Pathshala. So in this video, we will discuss all about effectors. So about the characteristic of the effectors, then about the types, then how, where and how it is synthesized, its secretion and translocation mechanism, and ultimately the function of the effectors. So first the definition. So the effectors are pathogen derived molecule. This is not derived by or from host plant or any other thing. It is derived from the pathogen itself. So pathogen only secrete the effector protein that or secreted by the pathogen into the host plant to promote pathogenicity or virulence by manipulating the host cellular processes. So main intention of producing effectors by the pathogen is to manipulate the host metabolism in favor of the pathogen. Okay, this is the mechanism. Next, the characteristics. So the effectors are hydrophilic in nature. Hydrophilic in nature means this effector do not anchor to the cell membrane, and that's why they are just secreted protein, not anchored to the cell membrane. Then the most important thing is they lack housekeeping function. Means if any mutation is there in the effector gene of the pathogen normal functioning will be not hampered. So it is not a housekeeping gene. This effector actually imparts virulence or pathogenicity for the pathogen. It is not a essential protein or gene for the pathogen. It's only means for disease causing. Next, it is coded by or encoded by AVR gene or A virulence gene. Okay. Then it determines host specificity of the at pathogen or species level. So this effector are the proteins that will act on a particular host. By that it determines the host specificity. Then how they interact? So two conditions will be there. One is in presence of resistance gene and another is in the absence of resistance gene. So the mechanism or the theory behind the interaction of effector with host protein is by the gene for gene hypothesis or gene for gene theory. So when see this diagram, so when the pathogen is there, it will secrete the effector protein. When the plant is susceptible, that case what happened, this effector will be not recognized by the R gene because in susceptible plant no R gene will be there. So this effector will successfully manipulate the host system in favor of the pathogen and ultimately it will cause disease. But in case of resistant plant, what happened? Resistant gene is there, so it will recognize the effector protein and it will trigger ETI, effector triggered immunity. Okay, ultimately defense system will be activated or plant will show resistance, not disease. Okay, but in case of susceptible plant, which system is triggered? Effector triggers susceptibility. Effector is triggering the susceptibility in plant in case of susceptible plant, but in case of resistant plant, due to the presence of a resistance gene, it is failed to triggering the susceptibility. Ultimately it is recognized by the R gene or resistance gene and further the effector trigger immunity is triggered. Okay, and defense system is activated and plant will show resistance. So this is about the interaction or how the effectors actually act inside the plant. So for susceptible plant, it acts as a virulence factor. It enhances the virulence, the pathogenicity of the plant. But in case of resistant plant, it acts as a virulence elicitor. I have already prepared a separate video on elicitor. Elicitor are the secondary metabolites or secondary proteins or compounds or the elicitor are the molecules that actually elicit or that enhance the plant defense system. So this in case the effectors, when recognized by the plant system or by the R gene, the resistance gene, they behave as elicitor. They will not call as virulence factor, they will call as a virulence factor or a virulence elicitor, because it triggers the defense, not susceptibility. But in case of susceptible plant, it will work unnoticeably because R gene is absent and it acts as virulence factor. So this is the major difference between effector and elicitor. As we all know that the elicitor are both biotic and abiotic origin. So the biotic elicitors are the pathogen derived molecules. Here the effectors are also pathogen derived molecules, but the only difference is how they behave inside the plant. If it is entering inside the plant and causing disease or triggering the susceptibility or inducing disease in plant, then it will be called as effector. But if that pathogen derived molecule, if it go recognized by the particular R gene and ultimately the defense system, if it is triggered, then it will be called as effectors. Elicitors. Okay. So this is about the effector and elicitor, its characteristics. Next, the effector R gene interaction. So I have already told that this effector are recognized by the R gene, and if they are recognized means ultimately the effector triggered immunity was triggered, and this recognition occur by gene for gene manner. So let's study this step by step. So effector are the product of pathogen virulence gene. This is true. Okay. And the plant gene product that recognize the effector is the R gene or resistance protein. So when this effector and R gene interacts, it will trigger the ETI. This is the gene for particular gene will be interacted with the particular another gene of the host plant. So how this recognition occur, whether this is a direct interaction or indirect interaction. Means direct interaction means the effector directly interact with the R protein, or indirect interaction means this interaction between effector and the R protein will be mediated by another protein. So let's discuss this. So three modes of recognition are there. First, the direct recognition. This recognition model is known as receptor ligand model. In this model, directly the effector will be bind to the R protein and they will recognize, and ultimately either the ETI or ETS, whatever is there. If the R gene properly recognize it, means ETI will be activated. If R gene fail to recognize the effector protein, then ETS will be activated. See, if any R gene is present, it doesn't mean that the plant will show resistance and it will activate the immunity against any effector. This recognition occur in gene for gene manner, very particular. So all the R gene can't recognize every effector. The interaction are very specific in nature. Okay. So this is about the direct recognition. Then the method of indirect recognition. So it includes two models, guard model and decoy model. So guard models means here this R gene actually not directly interact with the effector protein. So another guardy protein is there. Guardy protein, GUARDEE. So this guardy protein actually it guards the R gene. Okay. So this guardy protein actually it interact with the effector protein, and their interaction result in some modification in the guardy protein, and that will be recognized by the R gene, and ultimately it will be decided that whether the defense will be activated or plant will become disease or susceptible. Okay. Similarly, in case of decoy model, decoy the effector are interact with decoy protein, not the guardy protein. Decoy are actually not the guardy, means they are not the target of the effector. They are actually traps. During the evolution they are formed by the duplication of genes. Okay. So they just mimic the effector target. So by recognizing that interaction, R gene will ultimately activate the defense mechanism. So it's like the plant is putting some traps to trap the effector protein, and means plant is not allowing the R gene directly to interact this effector protein. They just set up some traps, and when that trap interact with the with our effector protein, that interaction will be recognized by the R protein, and ultimately the effector triggered immunity will be activated. So this is about the mode of recognition, direct and indirect mode of recognition. So next, about the types of effector. So based on location where this effector are located, based on its localization, three types of effectors are there. So type 1, apoplastic effector. So this apoplastic effector, present location is outside the host plant. So apoplastic effector location is outside the plant cell or in the apoplast, and the function is they function in the extracellular spaces including cell wall and intercellular spaces. An example of apoplastic effector is AVR2 and AVR4. Then cytoplasmic effector, the name only suggests that this will be located inside the cytoplasm. So it actually function in the cytoplasm and manipulated various cellular processes. A very important example is AVR beta. AVR beta is produced by Magnapatho rhizae. Next, the nuclear effector. Name also suggests that this will be located inside the nucleus. So this effector generally when enter into the plant cell, it will translocate into nucleus and modulate gene expression and transcription. Okay. Examples are AVR BS3 and this TAL effector. TAL effector are very important, transcription activator like effectors. So this is the effector present in BLB, Xanthomonas oryzae, Pathoba oryzae. This is very important. This actually the major virulence factor for BLB. Then so based on localization means after this effector enter inside the plant cell, where it localize and where it act. Based on that, three types of effectors are there: apoplastic, cytoplasmic, and nuclear. Okay, remember these examples. Okay, these examples are very important. So in this photo you can see the representation where this three types of effectors are located: apoplastic outside the cell, then cytoplasmic inside the cytoplasm, and in the nuclear effector inside the nucleus. Next, about the synthesis and delivery of the effector proteins. So how the pathogen produce and deliver the effectors into the plant cell. So generally the effectors are produced by fungi and gram-negative bacteria. It is not produced by any viruses. Okay, only produced by fungi and gram-negative bacteria, not the positive. Gram-negative bacteria and biotropic fungi. So in bacteria, it is having a special secretion mechanism, that is type 3 secretion system. It is a needle type apparatus. So what happened? The bacteria is having a needle type apparatus. So it directly inject the effector protein from its cytoplasm to the cytoplasm of the host. So this is also known as injectosome. Okay, type 3 secretion system, injectosome. Okay, this is found in gram-negative bacteria. So directly from the cytoplasm of the pathogen to the cytoplasm of the host cell, the effector protein will be secreted. Next, in fungi, the classical secretory pathway is followed. So this includes the endoplasmic reticulum, Golgi apparatus, and secretory vesicle. So from this endoplasmic reticulum to the Golgi apparatus, then secretory vesicle will encapsulate that effector protein and it will be ultimately released into the apoplast. Then from the apoplast, this effector will be enter inside the plant through various interaction, means endocytosis. Okay, so this is about how this effector proteins are secreted from the pathogen to the host cell. So in single word I can say, means in bacteria the secretion of effector protein occur by type 3 secretion system, and in fungi it follows the classical secretory pathway to secrete the effector proteins. Okay, so this is the secretion mechanism. Then examples of effector produced by bacteria and fungi. So example of fungi producing the effector are AVR4, AVR3A. Then fungal bacterial effectors are AVR-PTO, TAL effector. These things we have already discussed. So ultimately the final function of effectors. So how the effectors manipulate host to promote the pathogenesis. From the first slide, we have already discussed that effector are the pathogen derived protein that manipulates the host metabolism in favor of pathogen to cause disease, to induce susceptibility. Okay, so how this manipulation occur? So the first step is first one is suppression of PTI. So PTI is PAM triggered immunity or pattern triggered immunity. So here what happened? The PAM molecule, the foreign molecule, it will interact with the PRR, pattern recognition receptor of the host membrane. So when this PRR protein will recognize the PAMP molecule, they will trigger a basal level of immunity, that is known as PTI. So this effector actually neutralizes this basal level of immunity. Okay, this is the first step how this effector act on plant. Example is the AVR-PTO target FLS2. This is a PRR, pattern recognition receptor. So this pattern recognition receptor is not a big thing. These are just membrane proteins. Okay, so in the membrane of the host plant, receptors are there that are to recognize the, means at the entry level, at the entrance level, these are the guards that are recognizing the foreign molecule. Okay, this is FLS2. This FLS2 is also like that PRR protein. Okay, which is targeted by this effector protein AVR-PTO. Then second method how this effector acts is it modify the host metabolism. So this effector, what it does, so mostly the nuclear effector, nuclear localized effect. Three types of effector we have discussed: apoplastic, cytoplasmic and nuclear. So generally this nuclear effector, what it does, it enter or translocate inside the nucleus and it will alter the gene expression, transcription, translation, these things it alter. So by this it altered or modify the host metabolism. Examples are this AVR-BH3 TAL effector. Okay, so ultimately what it does, it actually rewire the metabolic pathway to enhance nutrient availability and support the pathogen fitness. Okay, so it will divert the nutrient to or for the growth of the pathogen, to support the pathogen fitness. Next, third is it suppress the defense responses. So first defense response it deactivated is PTI. This is the first mechanism. Other defense responses like the MAPK signaling pathway, salicylic jasmonic signaling pathway, ROS reactive oxidative burst. So these things also hamper, or this effector also interfere with these components. Okay, example is AVR3A of Phytophthora infestans. It suppresses the defense mechanism or defense response. Then final one mechanism is the effector alter plant structure. So it induces disease related morphological changes beneficial to pathogen. So some morphological or structural abnormality it may create. So the effector modulate the plant development and cell differentiation to create favorable niches for the pathogen. Okay, like this type of structure are formed by the pathogen that hamper the plant growth, but it creates favorable niches for the pathogen. Example is AVR-PTH by Xanthomonas citri. Okay, so this is all about effector proteins. So first we have discussed the definition of the effector. These are the pathogen derived molecules that promote the infection or that promote the virulence or pathogenicity by manipulating the host metabolic processes. This is hydrophilic in nature, lack housekeeping function, produced by AVR gene, act by gene for gene hypothesis. So by this act differently in resistant as well as the susceptible host. In susceptible host it acts as virulence factor, whereas in case of resistance host it acts as elicitor. Next, about the interaction. So three modes of interaction are there: receptor ligand model, then guard model and decoy model. Then about the types: apoplastic, cytoplasmic and nuclear. Three types of effectors are there based on its localization in the host cell. And the synthesis or delivery mechanism is the bacteria includes type 3 secretion system, whereas fungi include general or classical secretory pathway to secrete the effectors into the host plant. And ultimately the function of effector, so it suppresses PTI, modify host metabolism, suppress defense responses, and ultimately it alters the plant structure in favor of pathogen. So this is all about effectors. So we have already discussed the effector and elicitor. Next, what we will study, tell me in the comment section, okay? Otherwise I am thinking to make video on ETI, PTI, PAM, PRR. That will be better, I think. Okay, so thank you for watching this video.